Genetics is the study of heritable changes in the DNA sequence. When there is a mistake that happens in translation, then that mistake is translated and affects one protein. If, however, the mistake happens in transcription, that affects a subset of proteins translated from the transcript with error. What I mean by DNA error is a mutation. Mutations alter all proteins encoded by that gene and all proteins of all descendants of the mutant strain. Replication isn't perfect, so mutations arise during replication where DNA polymerase III is the primary replicating polymerase. Sometimes DNA polymerase makes a mistake or a mismatch, basically matches to a wrong bases. An additional mistake that DNA polymerase can make is slip forward or reverse when it goes over repetitive sequences and cause an insertion or a deletion. This happens at a much higher frequency than mismatch, but it requires the repetitive site. Mutation is also caused by mutagens. Mutagens are environmental factors that damage DNA. A few examples of those would be benzopyrene, which distorts the double helix, or methylnitrozoguanidine, which affects the chemical structure of bases. UV light can interfere with base cross-linking, and ionizing radiation can cause base elimination altogether. During growth, strands are segregated, and if a mutation occurs, then one of the daughter cells is going to inherit the mutation, and its daughters are going to inherit the mutation, and so on. So, mutations are heritable, and a mutant is a cell line that has inherited a particular mutation. The opposite of a mutant would be the wild type, which is a mutation-free daughter, or cell line, better said. Genotype. The genotype is the DNA sequence of a gene or a chromosome. Genotype is, for instance, the wild-type DNA, which gets transcribed into the mRNA and translated into a protein, and the protein represents the phenotype, which is defined as the measurable or observable trait conferred by a gene and mediated by proteins. An allele is a version of a gene. Each gene has two versions of itself, so there's a wild-type allele, which is the normal functioning version of a gene that produces a trait, and we have a mutant allele, which is a change in the DNA sequence relative to the wild type. There are a few consequences of mutation. Mutations change the sequence of a gene, needless to say, and this often results in a change of protein sequence. While changes in protein sequence change the protein shape, and this changes the function, and while a random mutation will almost always result in a protein loss of function, very, very rarely a gain of function mutation will arise, and we get to benefit from it. There are a few types of mutations out there, other than insertion and deletion and mismatch. There's missense, nonsense, frameshift, and silent. Missense mutations, single base pair change that changes the codon to a different amino acid. That changes the protein if it changes into a different amino acid. A nonsense mutation is a single base pair change that changes the codon to a premature stop codon, and that stops the protein right there, and it doesn't end up being what it was supposed to be. A silent mutation is a single base pair change that changes the codon but codes for the same amino acid due to degeneracy. That is one of the properties of the genetic code that is very useful when it comes to mutations, because if a base pair is accidentally substituted for something else, it is very likely that it will still code for the same amino acid, and that is good for us. A frameshift mutation is an insertion or a deletion of base pairs in amounts not divisible by 3, and this completely alters the subsequent amino acid sequence. Sorry, I need to get comfortable. My cats are taking the whole bed, so I'm trying to fit somewhere. Polarity is a side effect of mutations within operons. I was mentioning that some mutations can cause a stop codon to be inserted somewhere where it's not supposed to go, and that causes the row to bind and terminate transcription, and a mutation is created this way. Mutations early in an operon decrease or abolish the expression of downstream genes. So that's something to keep in mind. The most frequent mutations are those caused by mutagens. There are quite a few events per genome. Then there's the slipped strand mispairing, 1 per genome. Then we have 1 in about 1,000 genomes. We have loss of function, and the rarest of them all is a gain of function mutation, which occurs about 1 in 1,000,000 genomes. DNA has evolved to have repair systems so that mutation is minimized, because as you can see, it's not generally a good thing if you get a mutation. And there are 4 ways that I know of that a DNA repairs itself. A few lines of defense. We have DNA polymerase proofreading, we have methyl-directed mismatch repair, we have an SOS response and excision repair, and last but not least, a recombination repair. Let's talk about the mismatch error. Sometimes DNA polymerase makes a mistake, right? A mismatch, we've seen that. Well, DNA polymerase also has repair function. In addition to polymerization, it has a 3' to 5' exonuclease activity when a mismatch is detected, and it backs up one base and excises it. After excising mismatch, DNA polymerase 3 proceeds with replication, and that's it. But sometimes the DNA polymerase fails to recognize the mismatch and keeps going, and the mismatched base does not base pair correctly, and it causes a distortion. There's a second line of defense when that happens, and that is the methyl-directed mismatch repair. We have 3 proteins that I need to mention first: MUT-S, MUT-L, and MUT-H. So MUT-SLH. MUT-S recognizes and binds to the DNA distortion. MUT-L recruits the other protein I haven't mentioned yet, MUT-H, to the first one, MUT-S. It's a linker protein. And MUT-H is an endonuclease which gets rid of the damaged base. So the damaged DNA is excised, and the repair polymerase, DNA polymerase 1, reloads and fills in the gap. That's nice and everything, but how does repair recognize which base is the wrong mutated base? So how are mutations distinguished from wild-type sequences? Well, the older strand is presumed to be correct, so cells must identify which is the older strand. That's easy because DNA is hemimethylated, so the older original DNA strand is modified by methylation. The newer strand lacks this methylation, so MUT-H SL cuts out the distortion on the unmethylated DNA strand, as it has been more recently replicated. Easy fix. After replication, the DNA is fully methylated by an enzyme called DNA methyltransferase. What happens if we have damage after replication? How are mismatches repaired when a mutagen chemically damages its base, for instance? That is where the SOS system comes in. Let's begin with the protein called REC-A. REC-A binds to a damaged base and becomes activated, and it's called REC-A asterisk for some reason. Then we have LEX-A, which is a transcriptional repressor DNA binding protein that inhibits SOS genes. So REC-A asterisk causes cleavage of LEX-A and the repression of SOS genes. We see that in a few cases. In bacteria, we see it as an inhibitor of FTSZ, and that is named SOL-A. UVR-ABC is a DNA excision repair and polymerase IV error-prone polymerase. SOL-A inhibits cell division by interacting with FTSZ and blocking the Z-ring formation until DNA damage has been resolved. SOS system makes sure that DNA damage has been repaired before proceeding with cell division. UVRABC detects chemically damaged bases, and DNA polymerase fills in the gap. Last but not least, DNA polymerase 3 cannot replicate through damaged bases, so replication jams. And when that happens, as a last resort, DNA polymerase 4 is switched into replication fork, and it can polymerase past damage, but it has pure accuracy, so it's error-prone. However, they don't really get into that stage because of the first three DNA repair systems. Let's just review. We have DNA polymerase 3, which has the ability to back up and excise a mismatched base. Then we have methyl-directed mismatch repair, MUT-SLH, that can excise mismatches. And then the SOS, we have excision repair, UVRABC, that can excise damaged nucleotides, and the error-prone error-prone DNA polymerase 4 copies over the top of damaged nucleotides.